Welcome to the course on basic genetics. Three of the teachers, Dr. Richa Arya, Professor Madhu Gavaldas Tapadia, and myself Jagat Kumar Roy, a retired teacher from Department of Zoology, Banaras Hindu University, Varanasi, taking this course. This is week 11, human genetic disorders. In that, an introduction to cancer. This is lecture 4. We all know cells divide. Cells have inherent property to divide. They divide by mitosis. So this is a simple picture of mitosis where this is interphase, and then the chromosomes started condensing, prophase, and then they start showing the two chromatids. They are condensed well and they are getting organized in the equatorial plane. So this is pro-metaphase, and then comes metaphase where the chromosomes perfectly aligned to the spindle fibers. Then comes anaphase where the two chromatids of a chromosome getting pulled towards the opposite poles. The cell divides subsequently. Chromosomal division, once again, prophase, metaphase, anaphase, telophase, that we have already seen. These phases, the entire cell cycle, because this occurs in a cyclical manner, one phase after the another, again repeat the same phases. So that is why this is called as cell cycle. Just now we have seen mitotic phase, where we have seen prophase, metaphase, anaphase, telophase. Then comes the growth phase. After the telophase, the two cells divided. Then comes the growth phase. In growth phase, the first growth phase, the cells prepare for DNA replication. That is one of the important events for the cell, that it has to replicate its DNA. After this preparation, the S phase, where the DNA replication is actually occurring. Then comes the next growth phase, where cells prepare for the division. So here the chromosome condensation, the factors required for chromosome condensation and cell division, they become ready in this phase. The cell cycle, this is very well regulated through checkpoints. The first checkpoint, you call it as G1 checkpoint. Then there is another checkpoint, that is G2 checkpoint, and then the third checkpoint is M phase checkpoint or M checkpoint. What happens there? What the cells do? There are important questions those are asked. The important questions are, like in G1, the cell is asking question, or the molecules in cell is asking and answering. Past this checkpoint, if nutrients are sufficient, there will be certain sensors who will sense that nutrients were sufficient, will say yes. Then growth factors, that is signal from the neighboring cell whether it has come or not. If the signal has come, then the answer will come yes. Next question is whether cell cycle is adequate or not. If the cell size is adequate, that means the growth has gone on properly, and then the next question is whether the DNA is undamaged. If everything is okay, then finally it gets the signal divide. So that is how this goes from G1 to S phase. It completes S phase, that is synthesis phase. After that, second growth phase or G2, there again towards the end there is a checkpoint. This checkpoint has again several questions. Whether the chromosome replication is successfully completed or not. If replication is complete, then answer will come yes. The next question is whether there is any DNA damage. If no DNA damage, answer comes no, then the cell signals that okay, now it is ready for division. So this phase is progressed. Now it comes to dividing phase. In dividing phase, there is a metaphase checkpoint where there is a good amount of stress or strain that is evaluated on both side poles. If the chromosomes are attached with spindle fiber on both the side, then there will be equal strain on both the poles. If it is equal, that means the chromosome is properly organized, and then one should go ahead. There are molecules which guide which phase of cell cycle it should be. The two molecules which are very important, number one is cyclin, the second one is cyclin dependent kinase. When you have G1 cyclin, that is cyclin of G1 kind, then CDK in association with cyclin will phosphorylate only those molecules which are required for G1 phase. That means out of several proteins, some of the proteins will get activated, some of the proteins will get inactivated, so that finally the requirement of G1 is complete. Then there is cyclin for S phase. The cyclin name is because they appear and disappear. Then S phase cyclin appeared. This S phase cyclin along with CDK will phosphorylate only those proteins which are required for S phase. This way S phase will continue. Same way again the next cyclin comes, that is G2 cyclin, and with CDK other proteins will be phosphorylated and activated, and thus every phases of the cell cycle is done. So with this, let us have difference between normal cell and cancerous cell. The first and foremost thing is that normal cells are mortal, cancerous cells are immortal. The other thing is normal cells show contact inhibition, cancerous cells show loss of contact inhibition. Mortality versus immortality. The moment a cell or an individual becomes immortal, then they are not under the checks and regulations, and they can do anything. Same thing happens for the cell. If a cell knows that it has to die, that means there are regulations that are prevailing there and will not allow the cell to get deregulated at any stage. But in cancerous cell, they are immortal, that means this regulation is gone. Normal cells show contact inhibition. Whenever we come in touch with some object or with some person, we try to squeeze ourselves, or we try to little bit move ourselves away from there, so that we are not actually touching each other. Same thing happens for the cell, that there is contact inhibition. When the two cells come in contact, it knows that there is no space and it should not divide. In cancerous cells, there is loss of cytoskeleton, many such proteins which are associated with making a skeletal of the cell. When they are not in its normal condition, the result is two cells, whether it is touching or not touching, that is not sense, and thus the cell can divide, can divide, can go one after one above other and can keep on dividing. The term cancer, that is uncontrolled cell division followed by metastasis. Why such name cancer? The name has come from crab. Crab has, there are two things that is special for crab. One is movement. Crabs do not turn and go back. It always moves whichever direction it is present. It can move forward, it can move backward, it can move sidewise without changing its orientation. Cancer also, without changing its orientation, it can move from one place to another place. The cells from cancer move from one place to another place, and that is called metastasis. So, when we say the uncontrolled cell division, when we say cancer, the name has been taken from crab. The other thing, that crab, it has ciliate leg, and when it catches an object, if it does not leave, that the things cannot be removed from them. That means if it makes a bite, then if the skin is with the ciliate leg, and if the crab is not leaving, then the result will be either the leg will be broken and removed, or the skin will be torn and will get removed. So, these are the two conditions only. That means cancer does not leave in general. So, for these such kind of terminology, cancer is a two-step process. Step 1 is immortalization. Immortalization, as I said earlier, that if the cell is immortal, then it can become cancerous. So, number 1 is immortalization, and second is transformation, means further changes that can lead to cancer. If we go to causes of cancer, there can be genetic causes, there can be familial, that is running in the family, there can be sporadic, that means some mutation occurred in that cell that resulted in cancer. It can be epigenetic. Epigenetic means the genes are okay, but the regulation is changing because of promoter methylation, because of histone acetylation and so on. That means because of getting it more compact or getting it loosened, accordingly the gene function changes. The third is environmental, where you have physical factor, you have chemical factor, you have biological factor. Physical factors are high energy radiation. Chemical factors are certain chemicals like carcinogen, like mutagen. Biological, some such things like viruses. They are the cause of cancer. There are two types of viruses which have been associated with cancer. Number 1, the DNA virus. In fact, DNA virus was known early for its association with cancer. And some of the earliest known DNA virus is polyoma virus, means many type of cancer. So, this virus could cause several different kinds of cancer. Then many other DNA viruses were discovered, like adenovirus, like human papilloma virus and so on. On the other side, there are retroviruses. Retroviruses means they have RNA genome and they reverse transcribe after infection, so that they can get incorporated or they can make further copies of its genome, or they can express the genes and can cause cancer. The question comes whether these are why cancer they result in cancer. Is there anything they are benefited? The thing is, any kind of infection, those organisms should thrive successfully. Otherwise, they will die in the environment of a host cell. So, what they require? They require, first thing, that their replication should start. And if they have to start their replication, I am giving the example of DNA viruses, they do not have any special gene for causing cancer. But when they infect, the first thing, some of the genes get activated of them and they help the host cells genes to get initiated so that things become ready for replication and for cell division. The RNA virus or retrovirus, they have only 3 essential genes. In addition, there are the other genes which was associated with cancer, and they were named as oncogenes. So, here you can see a term proto-oncogene. Just now I said oncogene. Oncogene was from retroviruses or RNA viruses. A large number of oncogenes could be identified. Thus, at a time, the total cancer research was to identify different kinds of oncogenes. As many as 50 oncogenes were discovered at that time point. This was in 1970s. Then another one discovery that everybody has the oncogenes, that actually changed the cancer research to a different level. 2 groups, Varmus and Bishop from UK and from US, they were studying whether the normal people have some kind of the retroviral infection and whether they have oncogenes. They found everybody has all the oncogenes. Their further research was why such kind of a condition, how many infection. Then they could come out that these are not the oncogene from viruses, but we have the same genes within ourselves. So, we cannot call them as oncogene, and we started now calling it as proto-oncogene. What for they are present? Are they for cancer? No, they are for cell division. Some of the proto-oncogene products are growth factors. Some of them are growth factor receptors. Some of them code for intracellular receptor and intracellular signaling molecules. Some of them code for transcription factors. So, all these come under proto-oncogenes. If I show this diagram, it will be very clear that this is a cell, this is the membrane, this is the nucleus of the cell, and this is the membrane. On the membrane, there are receptors, so these are receptors. And a growth factor is bound with the receptor. So, growth factor binds with the receptor, result is signal transduction inside. When signal transduction, so this straight arrows, these are signal transduction occurring, reaching to the signal reaching to the nucleus, and certain gene expression occurs so that cell divides. So, this is the normal mechanism of cell division. If any time there is loss of regulation, then cancer may cause. You can see some of the inhibitors also. This is an inhibitor, this kind of line. So, this is an inhibitor, this is an inhibitor, this is an inhibitor. So, these are tumor suppressor genes. That means they do not allow the tumor to occur, the cell to divide. So, such kind of genes which are there, their products do not allow cell division to occur, and they are called as tumor suppressors. One of the important tumor suppressor is RB. RB name has come from retinoblastoma. What it does? This is present in the cell, in the nucleus, and this binds with one of the important factor E2F. This is one of the important transcription factor which helps in transcription of several genes required for G1 phase going on. If RB binds with this, this is not allowed to function, and thus this RB acts as a guard, not allowing cell division to occur. That means cell cannot divide ever? No, cell can divide. We did talk about CDK and cyclin and the different phosphorylation that go on. So, here you can see when phosphorylated, RB is phosphorylated, this leaves E2F and result is E2F is now doing all kinds of activities and result is cell division. So, this is a tumor suppressor gene mutation where RB is not functional, result is the retinoblastoma. Same thing is for adenomatous polyposis coli. Here also one of the tumor suppressor gene is deleted and that results into it. P53, another very important gene. Its product P53 is protein 53 kilodalton. It binds with the damaged DNA. Its first function is inhibition of cell cycle. The next thing is it immediately recruits the DNA repair proteins to make the DNA repair, and if DNA repair is there, then P53 leaves that place, it gets degraded and cell cycle continues. If DNA repair is not done, then it goes to apoptosis, that means kill the cell. So, this way every day lot of cancers like condition occurs and they are cured. So, this is just to show that normal cells, they are in a very tight, very well organized. Cancerous cells, they are loose, they shed and they go from one place to another place through blood vessels and through lymph, and this is known as metastasis. There are three main types of cancers: carcinoma, cancer of epithelial cells; sarcoma, cancer of supporting cells; leukemia and lymphoma, cancer of circulating cells. These are some of the example of the carcinoma. This is oral cancer, this is basal cell cancer. So, these are all epithelial cell cancers. Some example of the supportive cell, this is of the muscle cancer. So, these are sarcoma. So, with this I think the leukemia and lymphoma we will talk later. So, with this some of the discussion point, why such name cancer? Once again one should do this exercise, why? Can you differentiate between a tumor and cancer? We did not talk, thus you have to find out. Number 3, why it is such a deadly disease and what are the possibilities of its cure? Thank you.